Thank you for the opportunity to speak today about biologic interactions between HIV and SARS-CoV-2. I thought I'd start this presentation by laying in context the fact that we're dealing with two globally devastating epidemics. Up at the top is COVID-19, and these are numbers that are somewhat out of date now, but really reflect the location of many of the deaths that have occurred due to COVID-19 in the Europe and in the Americas. In contrast, if you look at the graph that's shown at the bottom, this is a summary of the global HIV deaths that have occurred over 40 years. And you can see that the levels, the numbers of deaths are orders of magnitude different, but also the location of many of the individuals who've died of HIV/AIDS is here in Africa or in Asia. So very, very different pandemics, but very much, unfortunately, particularly Africa, very much interlinked. We've been focused particularly on Sub-Saharan Africa, which is both under-vaccinated and bears an enormously high HIV burden. On the left-hand side here, you can see 7.5 million South Africans are HIV positive, and more than 2 million of these individuals are not virally suppressed. In parallel to that, South Africa and all of Africa really is being left behind in COVID-19 vaccination rollouts, as you can see in the schematic on the right-hand side, where in orange, down here at the bottom, you can see Africa trails the rest of the world dismally when it comes to vaccination rollouts for COVID-19. So we're in this unfortunate position where we have high levels of HIV-infected individuals in the context of a very low vaccine rollout. And for many reasons, that is problematic. There are many ways in which COVID-19 has impacted on people living with HIV. One of these is the impact of COVID-19 lockdowns on HIV care. This is a slide that was kindly given to me by Nigel Garrett, which shows the impact of COVID-19 lockdowns on HIV care, with many people reporting reduced patient attendances at healthcare facilities because people were apprehensive. We also saw a nearly 50% reduction in HIV testing and a nearly 50% reduction in ART initiations, though fortunately no marked change in ART collections. Of course, people living with HIV are also at increased risk of dying from COVID-19. This figure here, particularly the one on the right-hand side, shows you many risk factors that have been associated with increased mortality due to COVID-19. But if you look down here, particularly at the bottom, and individuals who are HIV positive, a twofold increased risk of dying from COVID-19 in South Africa. So not only are these people who are living with HIV struggling to access care, they're also at increased risk of dying from COVID-19. There's been a significant effort in the SARS-CoV-2 field, particularly in countries across Africa, to understand immune responses to SARS-CoV-2 in SARS-CoV-2 infected or vaccinated individuals, and particularly to understand that in the context of people who are living with HIV. And this is what I'm going to focus on in the next few slides. One of the first questions that we and others have been interested in is whether the vaccines, the SARS-CoV-2 vaccines, COVID-19 vaccines, are effective in people living with HIV. And there's several pieces of data, and I'm highlighting just a few here, and then we'll show you some immunogenicity data. But initially, to begin with, as we watch these vaccines roll out, great concern about whether these vaccines would show efficacy in people living with HIV. Part of this was triggered by data, for example, from the Novavax vaccine, which showed 60% efficacy in the overall population, but that dropped to nearly just below 50%, 49% with the inclusion of people living with HIV. The Chadox vaccine, also being tested in South Africa, was extremely well tolerated, with HIV individuals showing similar SARS-CoV-2 antibody levels. But important to point out that many of these trials, including some that I'm going to show you on the next couple of slides, are conducted in people living with HIV who are very well controlled, virologically controlled, and therefore not particularly immunosuppressed. And as I'll point out again later, this is a major gap for us. This is another slide that shows from the Chadox trials conducted in the UK, also comparing immune responses in individuals after vaccination, comparing people living with HIV with those who are HIV negative. On the left-hand side, you can see SARS-CoV-2 IgG responses, and you can see no significant difference between the HIV negative and positive cohorts. And just briefly alluding also to the cellular responses on the right-hand side, where again, no significant difference was seen. But as I've already mentioned, in all of our trials conducted globally, we've seen inadequate inclusion of people living with HIV, particularly with the focus on those who are immunosuppressed and therefore may have compromised immune responses to COVID-19 vaccines. Natural immunity, so infection-triggered immunity, is also compromised in people who are living with HIV. This is another slide from Nigel, which shows that there are reduced IgG and neutralizing antibody titers to natural SARS-CoV-2 infection in people who are living with HIV. And you can see here in a comparison of 70 people without HIV to 30 individuals with HIV, quite significantly lower SARS-CoV-2 neutralizing antibody titers. Similar work has been published by Alex Segal at ARI here in Durban in South Africa, who also compared immune responses using, in this case, the live virus neutralization assay in people living with HIV with HIV negative individuals. And you can see a significant difference in these graphs. High is good in terms of titer, and you can see that HIV negative individuals mounted significantly higher responses than people living with HIV. But again, to point out the role of viral suppression here, when you look at the frequency in panel B of non-responders, almost all of those non-responders, as you can see from this first graph, are amongst those who are people living with HIV. But when you partition those by whether they were virally suppressed or viremic, you can see that virtually all of the non-responders are in those who were viremic rather than those who were virally suppressed. Again, highlighting the importance of treatment for HIV positive people to restore their immune systems and enable them to react appropriately to COVID-19 and many other vaccines. We've performed similar work looking at a cohort of people living with HIV and who were hospitalized with COVID-19 in Steve Biko Academic Hospital. Very strong collaboration with Michael Boswell, Veronica Erkman, and others at the Steve Biko Hospital. What we did was we compared in wave 1 in South Africa, which was caused by the D614G variant, and in wave 2, which was caused by the beta variant, looking at individuals who were sampled at admission and 7 days thereafter. And we assessed individuals living without HIV in blue compared to those who were living with HIV, and looked at the magnitude and kinetics of antibody responses. This is work that was performed by Tumi Motsoneng, a PhD student in our lab. What Tumi shows is that people living with HIV mount similar neutralizing responses to HIV-uninfected individuals, whether looking in wave 1 or wave 2. So you can see here, again, the higher the values, the better the neutralization, and no significant difference between HIV-uninfected or HIV-positive individuals, whether in wave 1 or wave 2, in terms of either binding on the left-hand side or neutralization on the right-hand side. However, antibodies, we tend to generally consider antibodies in terms of neutralization, which is the Fab part of the antibody. But antibodies are much more complex, and there are many functions that are conferred by the FC portion of the antibodies, including antibody-dependent complement deposition, antibody-dependent cellular cytotoxicity, a relatively new function called phagocytosis, and complement and phagocytosis. What we did was we extended our studies of HIV-infected people beyond neutralization to examine each of these FC effector functions. The reason we focus on these is that FC effector functions, which are shown in the schematic here, are very much associated with reduced severity in COVID-19 infections and with increased vaccine efficacy. FC effector functions being associated with improved disease outcomes and with enhanced protection and therapeutic capacity of monoclonal antibodies, and in animal models certainly associated with vaccine efficacy. When we looked in the same cohort, again comparing HIV negative with HIV positive people across wave 1 and wave 2, again we see in general no significant difference between HIV negative and HIV positive. But if you look carefully, you'll see there are a few asterisks here that denote significance. In general, those are not between the HIV negative and the HIV positive, but are between different waves. So it does seem that the infecting virus, D614G versus the beta variant, seems to trigger slightly different responses. And this is something that we and others have previously published. So HIV infection per se here is not a problem. However, infection with different SARS-CoV-2 variants can trigger slightly different responses. The one thing that we did notice was although HIV negative and people living with HIV got to the same titers eventually, there was a slightly reduced kinetics in the development both of neutralization and phagocytosis. So there's ultimately everybody ends up at the same point, but there's a delayed response, particularly in people living with HIV. And this has implications for how we roll our vaccines out. I do note, however, that this cohort did not, again, include highly immunocompromised subsets of individuals living with HIV. And again, we don't know whether those groups of individuals will necessarily reach the same titers. All indications are that they probably would not. I'm next going to move on to the issue of long-term shedding and the potential emergence of variants. This has really attracted a lot of attention lately, particularly with the continued emergence of SARS-CoV-2 variants. And many studies indicating that HIV-infected people who are immunocompromised, along with many other groups of immunocompromised individuals, may struggle to clear SARS-CoV-2 infection, and therefore may represent a risk in terms of allowing SARS-CoV-2 to pick up additional mutations, which may result in the development of SARS-CoV-2 variants. This is data from the Ubuntu study here in South Africa, work led by Nigel Garrett and others, that shows that in individuals who are either seronegative or seropositive, the probability of a positive PCR is very, very much higher in individuals with low CD4 counts. And this is something that's consistent with many other studies, a strong correlation of CD4 count with asymptomatic carriage. We based much of what we know so far in individual case studies, but I think there's an urgent need to study this population further with systematic longitudinal sampling to understand exactly how often this happens and what the significance is. Certainly the case studies are fairly compelling. This is one example, again, from the Segal lab, that shows that HIV unsuppressed individuals may be long-term SARS-CoV-2 shedders. This was an individual who had a very low CD4 count, as you can see up at the top here, was infected with SARS-CoV-2 for 190 days, and during that time picked up quite a considerable number of mutations, including mutations we're now familiar with, the positions 501, 417, and others that we know to be associated with immune evasion. Characterization of that virus did indeed show that viruses that had evolved over long term in this individual were resistant at the top to neutralization by that individual's own plasma, and were also resistant to neutralization by plasma from unmatched individuals, convalescent donors, or from Pfizer vaccinees, highlighting the fact that these mutations had conferred resistance to antibodies. And there's several case studies now that support this. And this, of course, has led to much speculation about the emergence of Omicron, which now dominates globally. Omicron is, as you know, diverging into multiple different variants in South Africa. At the time of recording, BA4 and BA5 are pretty dominant. But all of these sublineages of Omicron all share many, many mutations, as you can see in this slide, spread across the SARS-CoV-2 spike. So each of these red dots here represents a mutation. And you can see they're often in the receptor binding domain or the N-terminal domain, which are known to be regions associated with immune escape. This is the most divergent variant that has been identified to date, though I should point out we've identified many others. And as I say, much speculation about the source. I'm sure we'll never know where Omicron and these other variants have come from down to the individual level, but the fact that we continue to have high numbers of individuals who are HIV infected who are not accessing treatment continues to represent a risk that we will see additional variants. And then in the last part of my talk, I want to briefly speak a little bit about translating the amazing success we've had with COVID-19 vaccines to HIV, and quickly address the question of whether we can translate our successes in COVID-19 vaccines back to HIV. And I think the answer is to some extent. It's very different. You know, we've had wonderful success with SARS-CoV-2, and with HIV, immunologically, it's a much harder target. I've summarized some here, and I'm going to show you in the next couple of slides a few more reasons why our success with SARS-CoV-2 does not necessarily mean that we have an easy path to an HIV vaccine. On the left-hand side, you can see with SARS-CoV-2, the immune response effectively clears the virus, whereas in HIV, the immune system cannot clear HIV and it integrates into our system. SARS-CoV-2, despite the huge hype that we hear about with regards to variants, is actually relatively stable, and I'll show you that just now, whereas HIV is highly mutable. Almost everybody develops neutralizing antibodies, good neutralizing antibodies in SARS-CoV-2 infection. Whereas in HIV, broadly neutralizing antibodies only develop very rarely in a subset of people after many, many years. And part of the reason for that is that the immune response to SARS-CoV-2 is highly focused, whereas the response to the HIV envelope is very much more polyclonal. I'm often asked, if it's so easy, why don't we have an HIV vaccine? If we could do that within a year for SARS-CoV-2 as a scientific field, why can't we do it for HIV vaccines? But It wasn't, in fact, a rapid process. We saw the emergence of these vaccines within a year, but they were based on 3 years worth of public-private partnerships and 8 years worth of coronavirus structural studies, and a good 15 to 20 years worth of basic biology, such as advances in platform manufacturing, nucleic acid vaccine development, and basic advances in understanding the HIV envelope structure. And 40 years, in fact, of RSV immunology and coronavirus biology. And so this notion that we have vaccines for SARS-CoV-2 very quickly is, in fact, not true. Again, comes to the biologic intersections between these two pandemics. Much of what we were able to achieve for COVID-19 vaccines were based on substantial amounts of work, often towards HIV vaccines. I mentioned that the variability in these two viruses is not comparable, and this is a beautiful slide from Will Fisher and others that really makes this point. Each one of these dotted ticks of color is a mutant. On the left-hand side here is HIV, and you can see that it's a swarm of different viruses, incredibly different from one another across the world. In comparison, SARS-CoV-2, despite the fact that we've seen these variants and despite the fact that they've caused such chaos, it's actually relatively trivial levels of evolution in comparison to HIV. I think really a sobering way of thinking about the difficulties of making an HIV vaccine still. We see insertions and deletions in SARS-CoV-2, and these are very important in HIV, but in SARS-CoV-2, they're relatively focused in location, whereas the spike of the HIV envelope can tolerate insertions and deletions in many, many more places. So it's a much more malleable protein. This is an example here of the one insertion shown to be important for SARS-CoV-2, but in HIV we see this the whole time, all over the HIV envelope. HIV is also almost completely naked compared to SARS-CoV-2. SARS-CoV-2's got many, many of these spike proteins embedded in the surface, whereas one of the ways in which HIV hides from our immune system is by sparsely covering the virion with envelope trimers. And that makes it much more difficult for us to see this virus immunologically. These are the comparisons of the two envelope and spike proteins, but you can see they're very, very different in terms of how easy it is for the immune system to cross-link two of those proteins. And then lastly, the HIV envelope is really extremely well hidden from the immune system by sugars, which are invisible to the immune system, as I'm sure many of you know. And so in comparison, the SARS-CoV-2 spike is really very exposed to the immune system and a much easier target. Certainly unclear to what extent those glycans might shift and change over time, but at the moment, it's a much easier immunological target. We've certainly seen huge progress in RNA vaccines for SARS-CoV-2, with vaccines such as the Pfizer and Moderna vaccines really making a remarkable difference to the SARS-CoV-2 pandemic. Are they the solution to an HIV vaccine problem? I don't think so. They will certainly help us to test new concepts in HIV vaccine pipelines much faster. But the intrinsic properties of the HIV envelope that I've mentioned in the past few slides remain a problem, even with the advent of RNA technology at a mass scale. So many, many biological challenges that still need to be addressed for an HIV vaccine. And that's where I'll end. Just briefly to summarize, antibody responses in people living with HIV in response to COVID-19 vaccination are relatively unaffected, but much more data is needed on vaccine responsiveness in viremic people living with HIV who have immunocompromise. The delayed clearance of SARS-CoV-2, particularly in people living with HIV and other immunocompromised groups, is associated with viral mutation and the risk of the emergence of variants. But the extent to which this happens is actually unclear, and we need many more systematic studies rather than relying on sporadic case studies to understand this better. And just a final reminder that HIV is a much harder vaccine target. And while COVID-19 vaccine progress will certainly enable us to move much faster, many of the challenges that are intrinsic to HIV remain to be addressed and are particularly important when we consider populations such as people living with HIV who really need access to good vaccines quickly. With that, I'm going to stop and thank my lab shown down here at the bottom, who've all worked extremely hard, have maintained HIV research program in the lab, despite the fact that many of our efforts were redirected to SARS-CoV-2, selected out many of the collaborators who've been key to much of the work that we've done, and our funders. And I'll stop there and thank you.